Actually, our next speaker for the day will be Dr. Vera Tang. And I'm extremely excited to introduce Dr. Tang. She's an adjunct professor and the facility manager at the flow cytometry core at the University of Ottawa. Dr. Tang received her PhD in immunology at McMaster University and has focused on research efforts on phenotypic analysis of viruses and extravesicular particles by nanoscale flow cytometry. Dr. Tang had become a widely recognized leader on submicron flow cytometry, and she has published extensively in the development of assay tools and protocols for the detection of small particles by flow cytometry. And we are extremely grateful that she has the possibility to join us today, virtually, of course, so that she can share her expertise on developing workflows for instrumentations and also data calibration for small particle flow cytometry. Dr. Chang's talk today will be on small particles flow cytometry, a workflow for instrument optimization and data calibration, I'm sorry. Please help me to welcome Dr. Vera Tang today.
Thank you. It's very, very kind. Can everybody hear me? All good? Okay. I am going to start speaking. Hold on a second. Audio looks good. Everything looks good. Sorry, I'm just seeing no response. So, hi everyone. Thank you for joining. I really appreciate being able to share some time with you guys to talk about something that I'm very passionate about, and hopefully will get you excited to do some of this research yourself. So yes, my series title is a workflow from instrument optimization to data calibration, but more often than not, I like to say it's about size, and size matters, but it's a little bit complicated, and more a lot of other things come as a result of it's complicated. So hopefully I don't scare you away, but get you more excited to do this research. So in the intro, yes, my day job is to run a core facility at the University of Ottawa. So that's probably straight north from you guys, not that far. Most of my core users are cell-based users, analysis, sorting, very standard stuff, 80% of them. And then I have a small population of researchers who are interested in small particles, and this is mostly extracellular vesicles and viruses. So people always ask me, what instruments I'm using for small particles. And the fact of the matter is, I think there's a lot of different instruments that can detect something. But how do we confidently detect something and reproducibly detect something is a much more interesting topic for discussion. But these are the instruments that we have in our core. Let me just change my pointer options. So yeah, we have a Fortessa, we have an SH800 for our sorting. For our small particles, we have our Beckman Coulter Cytoflex, and we also have an Aurora with an ESP. Right now we're doing some testing with the small particle detector, small particle flow cytometer, the Cytoflex Nano, and some of it is also done on the Cytoflex SRT. So these are two instruments that we're sort of doing some testing on to see how we feel about the data. Okay, so moving forward, what do I mean, or what do most people mean when we're talking about small particles? We're essentially dealing with particles that are in the size range of less than about a micron, down to, I guess, whatever your detection limit is on your instrument. And most of the time, for your standard conventional instrument, it's kind of somewhere around 100 nanometers for a biological. And that leaves us with a very interesting space for detection. We're looking at extracellular vesicles, we're looking at viruses, some bacteria, some marine viruses, plankton, phytoplankton, lots of cool stuff. And this brings us to the point, why do we want to use flow cytometry? I feel like the audience is probably sold on this technology, and I don't have to belabor this point, but why specifically do we want to use flow cytometry to analyze small particles? And I do hear people saying, oh, maybe flow cytometry is not the best technology for EV work or virus work, but hopefully I will convince you that it is actually quite great because flow cytometry is very high throughput. We can detect multiple parameters off of our cytometers. These detectors are very highly sensitive. We get very good resolution off of them, and it's a quantitative technology. So that's actually a very important thing. And I was sort of paying attention to the talks that we're going through earlier today, and I was excited to see that somebody else wanted to talk about quantitative flow cytometry. Excited to see that in other areas aside from small particle flow. But last but not least, I think flow cytometry as a technology for EVs and viruses is very important. The fact that we are looking at single particles, and that's at the heart of our technology, single particle detection. So just to look at some other single particle detection technologies that are used for EVs commonly in research right now, focusing on panel C, D, E, and F. B is obviously electron microscopy image of a sample. Now, this is a sample, unpublished data from a colleague of mine, Joshua Welsh, while he was working in Dr. Jennifer Jones's lab at the NIH. Now, this is a very interesting sample. It contains two different types of particles. It has a virus-like particle and it has an EV-like particle. And the nature of them is such that when you look at them in electron microscopy, you can see a difference. When you separate these particles out using a size exclusion column, you can see that they come out in different fractions. And when you look at the TEM data, the quantified TEM data, you can actually see that there are two populations, one smaller, one larger. Now, the interesting thing about this figure is comparing to the other technologies. So I apologize for the small writing, but on panel D here, we're looking at the microfluidic resistive pulse sensing technologies based on the Coulter principle. Down here on E, we're looking at nanoparticle tracking analysis. This is very, very popular technology. And last but not least, my favorite technology is, of course, the flow cytometry. So very easily, we are seeing the fact that we're taking transmission electron microscopy as our ground truth for analysis of the sample because it clearly defines the two populations. Resistive pulse sensing, not a bad job. It also picks up the two, but you can see the sensitivity is perhaps not as good as EM. And then looking at nanoparticle tracking, it seems to be the sensitivity is okay, but the resolution is not there. You're not actually able to separate those two populations. And the surprise and maybe not so surprising outcome is that flow cytometry does a very decent job of the sensitivity as well as the resolution. And if you look at the other benefits of flow cytometry, the fluorescence detection and the quantitative aspects of it, it ends up being sort of a level round good technology to analyze these extracellular vesicles. And hopefully I'll be able to convince you of that. All right, so those of you in the audience probably very familiar with flow cytometry. What is the difference between using flow cytometry for cells and EVs? And I get asked this question a lot. And I think that mostly to address this question, we kind of have to understand something about the limit of detection of our instruments and also the differences in detection sensitivities. So something that we as flow cytometrists, when we're looking at cells, don't necessarily encounter in an everyday basis is what is our limit of sensitivity or limit of detection on our flow cytometers? Because most of us are looking at cells, and these are well within the detection limit of our flow cytometers, our favorite instruments. However, when we're dealing with detecting small particles, these differences in detection sensitivities are more obvious because we're using our instruments at the limit of detection. Now, you can see that in all these cases, some of the small particles are not being detected, while we are always detecting all cells. So keep this in mind in terms of what we're going to discuss next, which is how do we then go about reporting flow cytometry data when our limit of detection is potentially different, and that directly impacts how much of the EV population we are detecting. So let's go back to something we're familiar with, which is PBMCs. So these are cells that are isolated from the peripheral blood. We are used to seeing the forward and side scatter. We've got a population of lymphocytes here, and then looking at very classic marker of CD4 and CD3. And then, of course, you have your CD4 positive CD3 cells. So naturally, most people, many people, will present the percent CD4 positive either in the lymphocyte population or maybe even against CD3. And this is perfectly okay. We've done this for a long time. This is how a lot of our reports, our data is reported. And so how do we do this, or should we be doing this with respect to something that you're not perfectly resolving? Because there's never this doubt that we're not resolving our T cell population, right? However, this is potentially happening when it comes to your extracellular vesicles. So how do we then deal with this? How do we then compare data from my lab to your lab, or even my lab over time if the instrument changes, right? So when not all particles are detected, and this population of particles that are not detected can change over time and definitely between instruments. So first thing we do is find some standard units. So find some ground truths, some standard units that can be used across all different instruments. And so just for some information, the standard units of flow cytometry, we don't often discuss it. We tend to like to talk in MFI, which is an arbitrary unit, for side scatter, which is what we use for flow cytometry of small particles, and fluorescence, which everybody should be familiar with in here. We have standard units that are of diameter, which is very easy to understand, and also of scattering cross section. So this is the case when a refractive index of the particle is not available. So fluorescence, these terms are probably more familiar to you guys: molecules of equivalent soluble fluorophore, MESF, equivalent reference fluorophore, ERF, and antibody binding capacity. So antibody binding capacity, in my opinion, has a lot more biological relevance because we're talking about how many antibodies are able to bind to a specific target. And that can translate very easily to biological understanding if you're looking at, for example, markers or receptors on viruses, as the situation of small particles. So I'm going to show you a little bit of data, and we're going to focus on size scatter, so mostly diameter data, and hopefully I'll convince you that the way we want to report, or the way we're suggesting that people should report, is going to be a more reproducible, concordant way of doing so. So this is data, very simple, scattering data off of cytometer 1. These are samples, platelet EVs, and RBC EVs. So these are commercially available. I think they are from Solarcus. And so we're just looking at them on cytometer 1. And if you guys notice the histogram here, the x-axis, they're not in MFI. So these have been calibrated to a standard unit of diameter, and we're calling this EV diameter based on an average RI. And I'm just drawing some gates. So this is easily performed on FlowJo. So when I draw a gate and I have a calibrated axis, this gate is very easily defined. I can find these statistics in FlowJo. I'm drawing a gate between 130 nanometers to 200 nanometers. And on this sample, I'm drawing a gate between 150 to 300 nanometers. So already you guys are probably starting to see where I'm getting at, because if I have a calibrated unit of data, I can very easily report all of my stats based on that unit. So another thing that I would like to draw people's attention to is the actual threshold. So in our workflow, we determine the threshold based on the noise, right? So if you cannot go below the noise, that's obviously your limit of detection, the wall. And we use a very clean buffer sample in order to determine this triggering threshold. So triggering threshold is here. And again, if we have these calibrated units on the axis, we can actually very easily quantify that limit of detection. You can hover over it, you can find a value. So now I'm showing you data on cytometer 2. So if you remember, the goal of reporting in standard units, of being able to quantify exactly how much of a population we're looking at, is so that we can compare data. Now, cytometer 1 and cytometer 2 have drastically different sensitivities. So you can see the limit of detection. So where that PBS, that threshold trigger is a very different value. And because I have calibrated units that are standard units, I can directly compare between the two cytometers. You guys could probably tell visually that, oh my gosh, there's so many more events in here. Now, because there is a standard unit, I can actually set the gates in the exact same place. I like to say that you're providing people with GPS coordinates to find those events again. But by doing so, you can do direct comparison between cytometers, between times, between very different cytometers as well. So I'd like to touch on another thing, which is to look at how you report that data with respect to percent positive versus concentration. And I know there's even outside of small particle flow, definitely in cellular flow, back in the day when I was doing my PhD, the researchers there were very interested in concentration as opposed to percent positive because they understood that when you're looking at your percentage positive, this number would vary a lot depending on what value you're using as the denominator. So in terms of cells, what are you selecting as your quote unquote cell gate will really determine the percent that you get. In this situation, we don't have a cell gate. In this situation, even in cytometer 2, where we have a much better sensitivity, you're still not detecting the full population of EVs. You can tell that a part of them are being cut off. Now, we go back to the fact that we can set gates in the same size range. Now we just look at these numbers and you'd be like, you very obviously, these numbers are never going to be the same because you're detecting less. And even if you're looking at the exact same number and you're detecting more noise in a more sensitive instrument, this value will change. And if you were to look statistically, the difference between these values, 19% versus 11%, even though we know it's not a very good way to do it, there's a huge difference in variability in the values. So how do we actually directly compare? So we are running these samples now on volumetric instruments. So it's very easy to get a volumetric count of the number of particles that you're looking at within a size gate. So I'm looking at these counts, and yes, there's some variability, but you have to realize that this is the same sample that was run on two different instruments, different time over the day. Maybe there's a little bit of variability, but if you're looking at those counts in the same volume, that there's significantly more concordance in those values, and the difference is a lot less. So you're able to reach this concordance much easier when you're talking about the same standard units. So data reporting. So aside from percentages, not using percentages and going with concentration, another thing that you should definitely think about is that size, that refractive index, and that limit of detection. So once you've calibrated your data, all of these things are probably easier for you to be able to report as a metric. So this is another example. This is samples run on the more sensitive instrument, cytometer 2. And these are just samples that we were looking at in the lab. Once again, we have our RBC EVs that were purchased, and I make some virus particles. This is a bacteriophage in-house. So once we've calibrated, you can notice that we are then able to determine the size, the median size of the different populations that we're seeing. We can report a limit of detection on these populations. Now, you'll notice that even though this is the same instrument, same settings, running the same PBS sample, this limit of detection is changing. And now this limit of detection is changing because size matters, but it's complicated. So that complicated part often is the concept of this refractive index. So if you look at this axis, I have actually calibrated using a different refractive index that is more reflective of the particle I'm looking at. So the refractive index of viruses, it will be a different value than the refractive index of EVs because they're made of different things. Viruses are more proteinaceous. They generally have a higher refractive index. So software is available to do this. We're not all just, we're not Dave Novo and able to generate our own software algorithms. I certainly am not. But there is available software for you to do this. And you will then be able to actually learn more about the biology of something as well, and the different physical properties of something just by looking at calibrated data. All right, so aside from that, you can always do comparisons. So you guys saw that I had different instruments in my lab. I often like to run the same sample, do the calibration, and kind of then look at the different limit of detections that we have and provide sort of ways for us to directly compare the same sample across the different instruments. Because I will have users who come in the core who are like, hey, Vera, I'd like to analyze this sample. Can we sort it? Can we sort the stuff that we see on the analyzer? And the fact of the matter is, most sorters are less sensitive than the analyzers that we have access to. So how do we find the population that we sorted? How does it compare with the data that they have from an analyzer? This is a way to be able to cross compare across different instruments. So this is a sample of extracellular vesicles that we make in the lab. They express GFP on the surface. And if you calibrate those instruments using the method that I'll be talking to you about shortly, you can then report in standard units of your axis. So in terms of fluorescence on the y-axis, we're doing MESF of GFPs and EV diameter on the x-axis. So these different squares that you're seeing are the different limits of detection across the instruments. I had a poster of this at Cyto in 2025. And so this gives us a very direct and quantitative method to compare performance of instruments. But why else would you want to do this aside from, you're a flow cytometry nerd and you want to know the limit of detection of everything? It has a lot of biological implication as well. So how do you apply this? This is work that Joshua Welsh and I did quite a few years ago now. But basically, we took a sample of a murine leukemia virus that I also generate in the lab, and we ran it on two different instruments, on the LSR Fortessa and also on our Cytoflex S. You can see in the uncalibrated data that yes, you have a population. You can sort of see in the Fortessa that part of that population is missing, but how do you actually make that quantitative comparison of how much of that population is missing? For those of you in the know, the LSR Fortessa and Cytoflex are very different instruments. Even the scatters are off of different wavelengths, different lasers. And so we're using same sample, very different instrument settings, PMT, APD instrument, and being able to compare this data. So let's get back to what I was saying. As opposed to just comparing data, what else can you get out of it? So the fact that you can understand the standard units of the fluorescence and the size of the particles that you're detecting actually gives you a lot of information about the biology. So what I'm getting at is this antigen density. So specifically for virologists who may be in the audience, if you can quantify the receptors on the surface of a virus, you can probably deduce kind of the infectivity of the virus maybe, right? Because if that receptor is responsible for entry into a cell, the more of them that you have, potentially the easier it would be for that virus to attach and enter a cell. So all of these observations with respect to EVs as well, maybe the density of a marker that allows these EVs to home to a certain area will be very interesting biologically. So calibrated units don't all just allow you to make academic and quantitative comparisons between instruments, but it will give you more information about the biology. So all this to say, how you guys are probably wondering, how do we do this? And generally, we've talked about converting to standard units, but exactly how, we will get there shortly. So before I jump into that, I just wanted to give a little bit of a primer on the difference between fluorescence and light scatter light and the conversion into standard units for these two. So converting fluorescence to a standard unit is actually very straightforward math. It's a linear correlation for the most part. And it's an assumption that we've made for many, many years. So in terms of arbitrary units, so that's what you're detecting on your cytometer, so the MFI units, and the standard unit is whatever you choose. So in this case, in the example, we have MESF, but we purchase MESF beads from vendors the vendor will generally provide you with the MESF value of those beads. In this case, there's 4 populations with 4 different intensities. You can draw a straight line correlation through them, and then your experimental will be somewhere on this line so that you can then deduce the value of MESF. It's very simply stuff that you can use Excel for to calculate. Light scattering calibration, or that correlation to a standard unit, is a little bit more complicated. Um, so your light scatter intensity is here on the y-axis, and your x-axis is your standard. So in this case, we've chosen diameter, and now you can see that correlation is not linear. This is not something that you can pick something to fit on Excel. It's a little bit more complicated. So it is based on me theory, that modeling that we're doing, and because it's a more complicated curve, you do tend to need more points, right, to fit that curve. Okay, so now we understand that. We understand that fluorescence may be easier to do on your own, scatter calibration, maybe not. So here we have the workflow. So the workflow utilizes software called FCM Pass. FCM Pass is currently free to use for academics, so please go check it out, download it, play with it, and see what you think. But essentially, this software, with the use of this software, you are able to implement our workflow, which we've documented in this paper, and the QR code to the paper is right here. What we're doing is helping you guys automate this optimization for instrument setting, maintaining instrument performance, because once you've done this, you'll know a lot more about your instrument performance. And I guess the goal, of course, is to quantify biology and be able to compare data across your different instruments. So this is the first figure in our paper. It goes through that workflow of what we're supposed to do, or what we recommend to do to get your instrument ready for small particle detection. So first thing that we like to do is looking at the cleaning QC. That's something that everybody in the audience who uses the flow cytometer is probably familiar with. The next part is to optimize our detectors. So generally, when people are looking at their cells, they will more often than not optimize their settings according to their sample. And nowadays, I think more and more people are familiar with the spectral platforms, which come with their settings that are already, the application settings that are already inbuilt and optimizing the instrument. So this is not as foreign a concept. So what we're doing here is optimizing the detector settings using some reference materials as well. So for the side scatter optimization, we generally use a small fluorescent polystyrene bead, and we do something which is, I think, close to or very similar to a voltration or gaintration. In our situation, we like to call it scatter DSI. So this is detector setting incrementation. That's just to be agnostic to either voltration or gaintration, depending on your detector. So basically, we trigger on fluorescence, and that's why we need the fluorescent bead. And then we run the full range of gain or voltage that is available on your detector on that instrument. So this workflow is essentially instrument agnostic. We've done it many different times. If you look up our paper, if you please do look up the supplementals, there's 88 pages of it, I think. All the data from the instruments that we've run the settings on, this detector setting optimization on, are there. So essentially, yes, we've got our setting optimization. Just to give you a look of what that data looks like when you pan through from 200 to 3000, you get some data. And essentially the algorithm FCM Pass is looking for the place where we have the lowest amount of noise. So that limit of detection, there's the lowest amount of noise, the settings that we want, and also wants to balance that with your range of detection as well, right? Because if you crank your settings too high, a lot of your events that you care about would possibly be off scale. Um, the next thing that we look at is setting that trigger threshold. So in this situation, in our workflow, I just want to comment that our strategy is to start with scatter triggering optimization. You may see other techniques out there which look at fluorescence triggering. So the same can actually work for this workflow. You would just start with fluorescence triggering as opposed to scatter triggering. So happy to answer any questions about that later. But essentially, we run our PBS sample, and we pick a threshold cut off at around 1,000 events per second on most of our conventional instruments. And now on the nano instruments, which run at a lower volume, we generally go a little bit lower in terms of events per second. So, okay, yeah, we do have a talk about here about talking point about the fluorescence triggering threshold. So the last but not, so the next step, we basically look at then how stable our event rates are. So your event rate may not be stable in this case because perhaps there still needs to be more cleaning done. So the example on the right hand side here is a situation where the event rate is fairly stable. A lot of the events, and this is running PBS only, are essentially sitting right at that noise, that trigger threshold. Whereas on the left-hand side, you can see that there's still kind of events over time that are coming through. I like to visualize that as things coming off the sample line. Maybe I just need to do a little bit more cleaning and troubleshooting. All right, so the next part in our workflow is to look at detector optimization on the fluorescence. And again, we rely on reference materials for this. We use multi-peak rainbow beads. Those of you in the audience who run cores or similar tasks will be familiar with these beads. There's multiple different peaks, intensities. This is just data of what that looks like. We again go through that gaintration. I think more people have probably done fluorescence gaintration or voltration than the scatter, but the technique is the same. However, we're looking for different things. So in this case, we're looking for the lowest instrument noise as opposed to separation of the bead peaks, because for small particle flow, we are looking at noise. We want to go as low as the instrument can go. So last but not least, oh, well, not last yet, we're talking about fluorescence calibration. So once you have all of the detector settings that you're happy with, an FCM pass will be the software. There's a modality in there which allows you to put all that filtration data in, and it will automatically do the analysis for you and spit out your optimal settings. So once you do that, you will be able to then run all your calibration materials. So these are the MESF bead for fluorescence or any ERF beads or any body capture beads. And when you have an instrument that is stably aligned, that you don't adjust the alignment every day, these calibrated materials, I can run them and cross-calibrate a rainbow bead and basically utilize that as my fluorescence calibration for all of the experiments that come until someone touches my alignment. So that usually happens when the engineer comes for a PM. And scatter calibration is similarly done. I run all of the NIST traceable beads to give me an idea of the calibrated values. And basically it builds that curve, which fits that me curve that you see. And one of the really nice things that comes out of calibrating your data in FCM Pass is it comes out with this report. So there's report when you do light scatter calibration. Something that you learn about your instrument is actually the collection angle. So it's an assessment of the alignment of your laser to that core stream and how well aligned it is. So we know at the anticipated collection angle if you know the geometry of your flow cell. So standard Cytoflex flow cell is a rectangular flow cell. You can ask the manufacturer for it. The collection angle is essentially going to be 55 degrees, or sorry, half angle of collection because of that geometry. So that full angle of collection is 110 degrees and half of that is going to be 55. So experimentally, just by doing this calibration, running those NIST beads across those populations, what we're able to predict is that this alignment on this particular instrument is 56 degrees. So that's pretty close to 55 degrees, which is the theoretical. So the other plot that comes out of here is that me curve with all the different populations of beads that I ran, and the assessment of how well these beads are fitting where we're anticipating them to be based on this model. So you can see that a lot of those populations are fitting quite well. If you're able to see the small writing here, the black line here is polystyrene, so the refractive index of polystyrene. Y-axis is scattering intensity in arbitrary units. So this is just whatever MFI you're getting off of side scatter. And then the anticipated, the diameter of the beads that you're using. The blue line is actually envelope virus. So this is the curve and correlation according to the fit of this me curve based on those polystyrene the beads. So you are able to determine the angle of collection, that curve, and because of that, you can then input different refractive indices to predict where your limit of detection is going to be, which is this line, for each of those different materials. So super useful if you're running a core facility and you have people who are like, hey, can I detect EVs of this size? And so all you have to do is do a scatter calibration, and you captured all this information about your instrument. So theoretically, I can see where this intercept is for envelope viruses. I can see where this intercept is for EVs, and of course the polystyrene here. So this red line represents that threshold that you set on side scatter that you did using your PBS sample. So something else that I'd like to just comment on, and hopefully I'm not running out of time, lots of things to talk about, is the fact that I notice a lot of people who work in small particle EV flow really like to determine their gate regions for their EVs using a set of beads, so a range of size. So it is something that's very easy to do. You draw your gates based on the population of beats that you have, and then you say, okay, this is my gating range. So there's something about doing that, especially with respect to looking at size and scattering data, that introduces error. So you cannot exactly repeat that. And this is why. So I'm showing you this me curve based on one instrument, cytometer 1. And you see this me curve based on cytometer 2. So if you see there's a subtle change in terms, and if you focus on that 450 nanometer bead population, what you're seeing is that even though that bead is 450 and it's run on both instruments, if you look at where 10 to the 6 arbitrary units is on one instrument versus the other instrument, sorry, it's actually in a different location. So this curve and the me curve, the different bumps it's like a fingerprint. It's unique to the geometry of your flow cell. It's unique to that alignment of your lasers to that core stream. So if you just draw a gate based on polystyrene beads on one instrument, um, it is not going to be directly correlative on a different instrument. Like you could probably maintain concordance within your own instrument, but if you want to take this data, if somebody wanted to repeat your data and find the same region, if they were to use the same strategy of gating on beads, you will not find the exact same region because this correlation is not linear. This correlation is unique to that instrument collection angle and that alignment and that geometry of your flow cell. So hopefully that answers a little bit. But getting back to our workflow, this is the last bit of the workflow, which is to finally begin your experiment. I've got some extra bits of reading here, and I'm also going to highlight this one reference, this framework for EV flow cytometry, my FlowCyte EV. So a lot of people, when they start off on this work, they're like, how do we start? Where do we go? And usually this is one of my first references that I send to my users to read. For me specifically today, we're going to focus on two aspects. One is the assay controls. So why that's important, we as flow cytometrists understand that you need controls in order to understand your data, specifically the assay controls to understand flow cytometry data of EVs and other small particles, pretty straightforward, and they very similar to what we'd be used to for cell flow. Specifically though, I'd like to point out three that may not be as familiar, of course, you'd be saying single stain controls are very straightforward, used all the time in cells, and 100% used for the same purposes in EV work as well, but we have an additional benefit. So people are always asking, how many different fluorophores, how many different markers can I put on an EV? What's the max number? And I guess the question is always it depends, right? Depends on the marker, depends on the expression level, etc. But your single color control is going to allow you to experimentally determine what is your max number. Because if your single color control staining intensity is the same as your multicolor staining intensity, then you know that there is no inhibition or competition of the different markers with each other. So this single color control actually serves double duty. The other two that I'd like to talk about and highlight is the importance of serial dilutions and buffer with your reagents, to check your reagents to make sure that you don't have a lot of unbound fluorophores or aggregated fluorophores. And this goes hand in hand with respect to how the instrument works. So this is kind of a very simplified image of a flow cytometer. You've got your core stream, you've got your lasers coming through, and this is a zoomed in view of the core stream. So 30 microns is pretty standard generalization of the width of the core stream. And then the line that you see across here is the blue laser. And generally we're just saying, you know, the laser height is probably around 10 microns. Might be different, slightly different depending on the instrument, but this is our example. And so you've got your 10 micron beam height across the core stream. You're looking at your cells, which is about maybe 10 microns or maybe a little bit smaller if you're looking at lymphocytes, 5 to 7. But when you're looking in terms of EVs, we're really talking about something much, much smaller. So this generally, you know, a average size EV or virus particle is about 100 nanometers. And I swear there is actually a dot there. It's just so small you can't see it. And when we're talking about then how much space in this 10 micron laser beam, some back of the napkin calculations here with some minor assumptions, and essentially you can do a lot. You can detect a lot of these particles in that amount of interrogation, right? In that interrogation space right there. And with respect to antibodies, you can do even more of them because they are definitely a little bit smaller in some cases than the EVs of interest. So all this to say, how do we know that we're looking at one particle at a time? Something else to throw at you and to contemplate is the fact that usually when you're looking for when you're doing cell analysis, you're not as cognizant of the fact that the reagent alone will give you signal. Unfortunately, and maybe unfortunately, once you optimize your detection settings for small particles, you start to see a lot more. In this example here, I'm just looking at antibody IgG conjugated to PE. On the left side panel is no antibody, just PBS. And then the next two panels are the antibody at 0.1 micrograms per mil and 0.2 micrograms per mil. I'm just running these as samples through my cytometer. You can see there's a lot of fluorescence detection just looking at that antibody. So a lot of times what I see is people think that this is true staining, this is true signal. The fact of the matter is the only way that you can actually prove that something is fluorescent signal and true single particle signal is by proving it and doing those serial dilutions. So here in this case, I did a series of serial dilutions and I'm only just showing you the 1 in 250 of that sample. So when we're talking about the actual concentration, the fluorescence signature changes. So this is how you know that the more concentrated sample was actually swarming or we were getting coincident detection. When you dilute and you see a shift in that profile, I can tell that the background here has come back down to where the PBS is. So I know that this is probably more true in terms of the actual single particles of signal. If you do a serial dilution of your fluorescent sample and it essentially looks the same throughout the dilutions, that's how you can tell. And there's ways to graph this quantitatively. So you graph your concentration versus the fluorescence intensity of the particles, and you should be able to see a straight line correlation, right? So the other part of this MyFlowSite EV framework is obviously the calibration of instruments and data acquisition. And we already went through some of this earlier today. And I do hope that you guys get a chance to look at this later on. And definitely, if you have any questions, please feel free to reach out to me. And I believe I am out of time.
Thank you, Vero. Thank you very much. Okay. So we have time. We have time for two questions. One from you guys, if you have any questions, and one from the Zoom. Do we have any questions for Vera? Apparently, not. Oh, we got one down here. Thank you.
Hi, Vera. Can you hear me?
Yes, all good.
Okay, great. My question is simply for that last set where you're doing the serial dilutions, do you start or do you try to start the serial dilutions to have like 1 particle per unit volume per second or something around there, or do you go that far down?
Actually, not as detailed as you're suggesting. So it really depends on how much the user knows about their sample. So if I am the one creating the sample, generally I have sort of a ballpark idea of what that concentration should be. But users will generally many of my users use NTA. And so they kind of usually have an idea of ballpark concentration. So down to maybe 10 to the 9 particles per ml or 10 to the 8. And usually we do tenfold dilutions down to the point where I should not be detecting anything. So usually on the Cytoflex, just the standard Cytoflex, on the Aurora, on the Fortessa, so your standard cell instruments, I aim for something around 10 to the 6 particles per ml. That said, sometimes it really depends on the particle on the sample, because if you have a really heterogeneous sample, it's actually quite hard to predict concentration, and you will have to go down further. So if you think you're somewhere between 10 to the 5 and 10 to the 4, start with those lowest concentrations and run them and see what you get. And essentially, the workflow is they don't even bring a stain sample until we've had a look at their reagents. So as you saw, that antibody there was definitely swarming on its own. So we do a serial dilution of the antibody to determine kind of the maximum concentration that we can run on that sample. Because one thing that is important to note here, a lot of the workflow when it comes to looking at labeled EVs is to not wash the sample. So you stain at a high concentration of EVs with the antibody, and then you dilute to run it on the instrument. So there is kind of a maximum concentration of antibody that you can run, and then the way you adjust how much EVs, the concentration, the staining concentration sometimes can be you adjust the amount of EVs that you have, because sometimes you have a really, really dirty antibody and you really can't go higher than a certain concentration. So the only way that you can sort of improve the staining is to reduce the number of EVs or maybe increase that stain time and that sort of thing. So it's kind of a slightly different approach to optimizing staining when it comes to EVs and small particles, because you have limitations in terms of how much antibody you can use.
Let me go to Chris. Chris, do we have someone from online? No, that's great. And we say thank you to Vera. Thank you so very much. Thank you.